Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Optimizing Low GWP pMDI Sprays for Enhanced Performance and Sustainability. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Kindeva, who developed the content for this presentation. Kindeva is a global contract development and manufacturing organization focused on drug-device combination products. They develop and manufacture products across a broad range of drug delivery formats, including pulmonary and nasal, injectable, and transdermal. Their service offerings span early-stage feasibility through commercial-scale drug product fill finish, container closure system manufacturing, and drug device product assembly. I'd now like to introduce our speakers for today's event. Ben Myatt is a senior product development engineer within R&D at Kindeva Drug Delivery. His work focuses on next generation low GWP propellants, evaluating and developing current and new inhalation technologies and device platforms, and developing new test methodologies and novel apparatus to measure, understand, and enable new pMDI product development. And Daniel Duke is a senior lecturer in the Department of Mechanical and Aerospace Engineering at Monash University Clayton campus, where he leads multiple active Australian Research Council projects. His area of research is turbulent multi-phase fluid flows, and he specializes in the development and application of synchrotron X-ray diagnostics, optical diagnostics, and high-performance computing tools for studying these problems. And so now, without further ado, though, I'd like to go ahead and hand things over to our first speaker for today. Ben Myatt, you may begin when you're ready.
Hello, Ryan, and thank you for the introduction. As Ryan has told you, today we'll be talking about optimizing spray formation of low GWP metered dose inhalers, focusing on unlocking product performance understanding. I want to recognise collaborators from Kindeva on this particular slide, Phil Cox and Steve Stein. They've both been heavily involved in this research project, which we'll discuss in a few slides' time, as well as Dan, obviously, as the lead academic.
Thanks, Ben. I'm here as the leader of the multi-phase flows research group from Monash University. We're located down in Melbourne, Australia, so on the other side of the world from where many of you will be. Monash University is a research-intensive institution. We're a top 50 university with a very strong focus on medical and biomedical research. My group in the multi-phase flows area in mechanical and aerospace engineering specializes in the development of methods to measure very complex multi-phase flows. And this is a picture of me with some of my PhD students, postdocs, and our very enthusiastic honours students who all work in this area. And we are delighted to be working with Kindeva to accelerate the development of low GWP pMDI products in particular. Thanks, Ben.
To start with, I'm just going to get everyone centered on pMDIs, what they are and how they work, just so we're all on the same terminology. A pMDI is a pocket-sized active device delivering a precise dose or puff of medicaments to the respiratory tract for treatment of respiratory diseases. They're powered by liquefied propellant gas that flash boils and atomizes when the canister is depressed. That liquefied gas formulation is contained within a canister that has a crimped valve that meters a precise dose of that formulation upon every actuation, every puff. And the dose is emitted via a plastic actuator, which sometimes contains a dose counter or dose indicator. And there's really two formulation paradigms for pMDIs. One is a suspension of the API particles contained within that liquefied propellant, and sometimes we add some excipients to either aid the physical or chemical stability of those particles. The second paradigm is a solution of the API that's dissolved in a co-solvent and the liquefied propellant. Again, we may also add some excipients to aid the product performance or stability characteristics. On the right-hand side, you can see a little schematic of the cut through, showing the key components within the device. And another point to note is that this isn't the first time we've gone through a green propellant switch for pMDIs. The Montreal Protocol in the end of the 1980s drove the switch of CFC-based propellant gases, changing to HFCs or HFAs, hydrofluorocarbons or hydrofluoroalkane gases. And that kind of continued throughout the 1990s and into the early 2000s. And that was driven by the fact that CFC gases contributed to depletion of the ozone layer in the stratosphere. Today's green challenge is driven by the Kigali Agreement to the Montreal Protocol, which is identifying the phase down of emissive uses of hydrofluorocarbons or hydrofluoroalkane gases with the intent of slowing global warming. The current pMDI propellants, HFA 134A and HFA 227, both have relatively high global warming potential values, and these are relative to emissive use of CO2. Two alternative propellants that are being explored in the industry at the moment, HFA 152A and HFO 1234ZE, as you can see on the slide, have much lower global warming potential values. And of course, the legislation that backs up this, the Kigali Amendment and enforces the commitments to the Kigali Agreement in the different regions is currently being developed. You can see on the slide a few of the different regional regulations that are at play, and they're currently being worked. The second fact is that the economics of propellant use is changing. The major pharma players have publicly stated that they will be switching to low GWP propellant gases in the region of 2025 to 27. And Cora, one of the main suppliers of HFA 134A in industry, has heavily invested in development of HFA 152A, which is helping to drive the transition. Finally, medical grade HFA 227. The propellant price of this has really risen quite dramatically in the past few years, and we expect that HFA 134A is likely to follow. And this is really driving the switch to pMDI propellant gases to low GWP alternatives. And of course, the first question we have to ask is, well, what does this mean for our product performance? So if we look at the properties of these new propellant gases, we want to try and understand some of the fundamental aspects of these gases and their use in pMDIs. And we know there are some significant differences in the thermodynamic and physicochemical properties of these propellants. For instance, the density of 152A is significantly lower than the other gases that we can see on the slide. And that will have an impact on the physical suspension stability, for instance, of a pMDI product. You can see that the surface tension values of HFA 152A and 1234ZE are also higher than the current gases, and that will have an impact on the initial droplet size that is formed upon atomization, and then subsequently the final residual droplet size that will get into the lung. The thermodynamic properties of 152A are also fairly different to the other gases, being outliers, and that is going to impact the atomization process, probably hindering the process along the way. And that's also evidenced by the Jacob number and the gas phase Weber number. These values are much lower than those values for the current gases, and this really just tells us that there is less energy available for the atomization of the gas into respirable droplets. The atomization process will be less aggressive. But of course, we don't understand just by looking at a table of properties what the impact on product performance will truly be. Ultimately, however, the bottom values of this table, the global warming potential, are the reason that we're doing this work. And of course, you can see the values for the new gases are significantly lower than the current gases. So we introduce at this point the collaboration between Kindeva Monash University and the Woolcock Institute via the Macquarie University in Australia. This is a joint research project funded by Kindeva and the Australian Research Council. And we're really seeking to leverage the capabilities of a really technically strong, world-leading team that's cross-functional within Kindeva, the pharmaceutical knowledge, Monash, the mechanical engineering and thermodynamic property knowledges, and within Woolcock and Macquarie, the pharmaceutical translational sciences. We're using advanced techniques to facilitate our fundamental understanding of the physics behind pMDI atomization and how those processes change when we switch to the low GWP propellants, with the ultimate aim of enabling new products with low GWP propellants. We're going through 3 phases of this project. Initially, we've undertaken some benchmarking to simply understand what differences we're up against, what propellant property differences, how they're impacting the product performance of pMDIs when we switch. We're undertaking investigations into mitigation strategies and how we can optimize performance using our current hardware and current formulation toolboxes. And then we're moving on currently to new opportunities. So how can we push the envelope of pMDI performance and maybe new novel hardware developments? We're going to look at the challenge of pMDI performance analysis. The aim here is to predict the role of the formulation and the actuator on droplet and ultimately particle outcomes. But it's worth saying that the atomization process of a pMDI is a highly complex, transient process, both inside the device and in the spray itself. The liquefied propellant gas undergoes a rapid depressurization when the pMDI is actuated and the patient receives the puff. That liquefied gas starts to flash boil and produces a vapor liquid 2-phase mixture as it passes through the device. On the right-hand side of the slide, you can see an X-ray image that Dan's group have produced, looking inside the actuator as the fluid flow is taking place. You can see a highly chaotic and transient fluid motion that is very difficult to predict. When you then start to look into the spray plume itself, again, this flash atomization process that produces the droplets is highly chaotic, highly transient and turbulent, and difficult to predict in many respects. Of course, spray to spray, shot to shot, the propellant behaves differently as the droplets evaporate. And we need to understand that initial atomization near the orifice where we produce our droplets, and then subsequently how those droplets evaporate and evolve as they mature into particles that can be respired by the patient. We can also see on the right-hand side figures that the effect of the co-solvent, in this case ethanol, has and other excipients on the internal fluid flow structure. You can see that as ethanol concentration is increased, the fluid flow structure changes quite significantly into a bubbly flow that's got very small bubbles. And this fluid flow structure right before the orifice has a big impact on the atomization of the fluid into small respirable droplets. It also has a big impact on the spray plume characteristics. And we know this for 134A gas and 227, but what we don't know is how changing to the low GWP propellants impacts the performance of pMDIs. So the first phase of our work was to understand how, if we use simply to change the propellant in the pMDI product, the performance changes. So we've formulated a range of formulations, as you can see on the slide, ranging from propellant only through to differing ethanol concentrations, to understand if we simply change the propellant but remain the same hardware, the same formulation recipe, what performance differences do we see? Where do the shifts occur? We've employed standard pharmacopoeial techniques, NGI and ACI, and maintained our componentry from Kindeva. So on this slide, you'll see the APSD results. And we highlight here in the first panel, the pure propellant only BDP formulation. We can see that the fine particle fraction of the 152A formulation is much lower than 134A, and both are lower than the 1234ZE formulation. Concurrently, the actuator mouth-throat deposition follows the same trend, in the sense that 152A has the highest mouth-throat deposition, which evidences why we get the lowest fine particle fraction. And if we add in some spray pattern area data, we can see that the 152A product has the widest spray pattern area, which suggests that the reason for this trend is because of impingement of the spray on the mouthpiece and into the throat. As we add more ethanol to the formulation, we can see that the differences between the different propellant product performances start to diminish. Ultimately, when we get to around 15% weight ethanol for this particular product configuration, we can see that the performance of the different products has converged. We can also see that the spray pattern area of the 12% ethanol formulation starts to also come closer together. So we can see that the addition of ethanol masks the performance differences we see between propellants. And we can see that the propellant itself does have a performance impact on the product that we're formulating. But from these cascade impactor data, we really don't have much information as to why this is the case. A lot of the temporal and transient information about the atomization process is masked by the cascade impactor measurement method. So we have a lack of understanding and knowledge as to why we see these differences. What we really need to do is be able to understand how the vapor pressure, the density, the surface tension, heat capacity and latent heat of these propellants impact the product performance. We want to understand how these values or these properties impact the droplet size variations we see and the plume structure and the dynamic effects that we see in the plume. So moving forwards, we've expanded the scope of our study to include a salbutamol suspension product, and we're maintaining all three of these propellants. We've undertaken a couple of other pharmaceutically recognised and typical measurements to try and understand if we can see more trends. So Malvern Spray Tech data for saltamine diameter, otherwise known as D32, is a drop size metric widely used in the engineering world, as well as the mass median aerodynamic diameter for both our salbutamol suspension formulations and our BDP solution formulations. And although it's difficult to kind of see gross correlations, what we can see, the broad trends are that the droplet size increases when we move from a 134A current product to a low GWP propellant formulation. However, even if we look at the drop size distributions behind these bars, it's difficult to understand why these trends are the case. Moving to look at the matured particle morphology by scanning electron microscopy, we can also see that by changing the propellant in the formulation, we see differences in the matured particle morphology. We have a different ratio of smooth spherical particles to corrugated rough particles. And again, we don't understand fully why this is the case. So far, we've seen that by changing to low GWP propellants, we've seen increased droplet sizes. There's a mixed trend with the fine particle fraction and the fine particle dose or fine particle mass that's measured. And we have a variation in the particle morphology. We understand that the atomization process of a pMDI is highly complex, and there's no real clear correlations in the data that we've showed so far from our initial benchmarking study, and that's going to unfortunately lead to poor predictability. So we have undertaken a series of experiments with Monash University and the Woolcock Institute that Danny's going to talk about now that enhance our understanding of the atomization mechanism and the droplet evolution processes of these low GWP propellant formulations, with a view to being able to then take that learning and understanding to tune our products going forwards.
Thanks, Ben. So our group at Monash University is interested in studying how the behaviour of the spray plume is impacted by the switch to a low GWP propellant and initially we're looking at this in matched hardware. So we will hold all conditions constant and simply change the propellant and the formulation and understand what that does. But to truly understand how the propellant alters the plume and thus alters the pharmaceutical outcomes that Ben's just been talking about, requires a measurement precision inside the spray plume, which is simply beyond anything that's currently possible with any of the conventional measurement techniques, such as some of those that Ben's been discussing. And so we've developed an entirely new ultra high speed imaging facility at our laboratory in Monash University, which goes well beyond the resolution and capabilities of any spray pattern and plume geometry system that's currently available on the market. We collect extremely large volumes of data and mine that data in order to extract the differences and the underlying drivers of difference between propellants. So shown here on the slide is a simple top-down schematic of our experiment. We use a hundreds nanosecond pulsed LED light source, which is custom built to produce a very powerful pulse of light, which is then travels through the spray. Droplets and particles in the spray scatter that light and absorb some of it. And the transmitted light is picked up by a very high-speed camera running at over 100,000 images a second. And this allows us to capture the dynamics of the spray. And we do this both inside the actuator and outside. The panel on the right-hand side shows you the various imaging regions that we've obtained by cutting an access window into the mouthpiece. In order to ensure repeatable conditions for our experiments, we also developed a pneumatic shake, hold and fire system, which allows us to repeatably shake and fire the actuator while maintaining precise airflow, humidity, temperature, etc. And to be able to do this over hundreds of actuation events. And this results in a large data set on the order of about 10 terabytes of data across the various propellants. And we then take a big data approach to extracting the underlying statistical variations between the various sprays. Shown on this slide here are some movies straight off the camera of plumes from our different propellants. So on the left column here, we have our solution formulations, and in the right column, the suspensions. And from top to bottom, we have our propellants, our HFA 134A, which we consider a control condition, our existing product, HFA 152A, and HFO 1234ZE on the bottom. It's very difficult to tell what's going on in these sprays from instantaneous images because they are so turbulent and complex. In fact, turbulent mixing flow inside the plume is one of the great unsolved problems in physics. So this really is a very difficult problem that there is no exact solution to. So in order to deal with this, we collect very large amounts of data in order to mine it for that underlying information. So we collect hundreds of spray events over time, and we then clean those images up and process them by removing the background information to extract what we call an extinction profile of the spray. This shows us where light has been absorbed and scattered by the droplets and particles at about 30 micron resolution at over 100,000 images per second. And from that database, we can then perform some statistical analysis. So for example, we can calculate the average or mean extinction profile, and this shows us the underlying structure of the spray behind all that turbulent mixing. We can then also look at higher order statistics like the standard deviation, which tells us how much variation about the mean we are seeing in a single spray plume or across multiple spray plumes. So here we have a test data set where we're looking at our solution formulation and we're simply varying the co-solvent concentration for HFA 134A. And this is a formulation we understand. We can see on the top row here that the mean extinction, the average value, really does not change very much as we alter the properties of the formulation, but it's in the standard deviation on the bottom row where we see the more significant differences. And we can see that as we expect, when we go to higher ethanol concentrations, we get increased core extinction, so there's more liquid present, and therefore this tends to cause a stabilizing effect on the spray. There's less turbulent vapor and mixing. So this allows us to sort of benchmark the technique. So now we'll move on and actually apply this technique to our low GWP propellants, and we'll have a look at what we get. In order to analyze the performance of low GWP propellants, we have to ask some specific questions. And the two questions that we're asking when we're mining this data set are, firstly, how stable are the sprays? And secondly, how repeatable are they? By stability, we mean how much variation over time do we see within a single actuation event? For repeatability, what we mean by this is how much variation do we see from one actuation to the next actuation? How repeatable are they? How much variation in targeting or spray plume width, for example, do we see? The diagrams on the bottom give you an idea of why these two things are important. In bottom left, we have a spray device that's giving us a very stable and repeatable plume. The extinction profile from our measurement will come up and then go down again over time, and it'll do the same thing every time. In the middle panel, we have a spray that is stable, but not repeatable. That is, it's relatively smooth, but it comes in a different profile every time. And so we're not always going to be delivering the same dose. On the right panel, we've got a spray that is quite repeatable. It's very much the same every time, but it's unstable. It has poor stability. And we see that in these large wobbles over time. This is going to increase the likelihood of deposition on the mouthpiece, for example, as we're getting more turbulent mixing and more chaotic behavior in the plume. So first, we're going to look at our BDP solution formulations. These are the time average results for the behavior of the plume inside the mouthpiece at very small scale. Here we're looking at about a few millimeters, and you can see that on the left panel we've got our 134A, in the middle 152A, on the right 1234ZE. And then these really aren't, there's not much to say about these, they all look very similar. But when we dig into the underlying statistics, then we start to see the differences. So shown in the middle panels here, this is the repeatability. How repeatable is the spray plume from shot to shot over hundreds and hundreds of shots? And what we see here is that the HFA 152A is a slightly higher value, a bit more red there in panel E, and this is telling us that it's a little bit less repeatable. The most stark differences, however, come when we look at the stability, and that's in these bottom panels. And here we can see that the HFA 152A formulation gives us a much higher stability measurement, and what that means is that the spray is actually much less stable. It's moving around a lot more. Why is this so? Well, HFA 152A is lower density and relatively high vapor pressure. So there's more opportunity for mixing. With that lower density, it's going to be pushed around more by entrainment of air into the plume. And this correlates with some of the actuator and throat deposition measurements that Ben showed earlier. Here we have the results for some salbutamol sulfate suspension formulations. These behave similarly, but we now see that the effects are simply enhanced. So we're getting redder and higher levels because now we no longer have the masking effect of the ethanol. The standout here is, as indicated by the arrow, is our HFO formulation has a much lower repeatability value. What this means is that the spray is much more repeatable from one shot to the next. The spray core is narrower and it's more dense with a lower vapor pressure, and therefore there's a slower mixing process occurring here. And this gives us less variation. It's neither good nor bad, but it's simply a design change that we have to account for when we're looking, for example, for equivalency. So if we want to understand how these different effects that I've shown will ultimately impact, for example, mouthpiece and throat deposition or the delivered fine particle fraction, we need to get more quantitative. And we need to try and find a way of ranking and comparing the performance of these different sprays. And one way we do this is by volumetrically integrating all the light extinction within the cone that bounds the spray plume. We call this the volumetric extinction. The value of this number on these graphs here really is not important. What is important is how the different propellants, indicated by the colors, how they vary. And what we see here is that in the left panel, which is the flow very near to the orifice, we see that the blue line, HFA 152A, gives us much higher values. That is, it's spreading much wider. And the HFO 1234ZE gives us slightly lower values. The plume is narrower. And this corresponds to the thermodynamic state of the fluid when it leaves the orifice. The 152 is wider. The 1234ZE is a narrower, denser plume. On the right panel, we can see what happens when these plumes then exit the mouthpiece. And while the 152A propellant continues to expand and maintains a high value, the 1234ZE formulation catches up with the 134A. The black and red lines come together on the far right side of the screen there. And what this tells us is that as the evaporation is completed, the average mixture density of our HFO formulation starts to come closer and closer to the 134A formulation, and they come into equivalence at some distance downstream. So we can take the same approach, and we might now ask the question, what region of the actuator is actually, or what hardware design variable is actually influencing this behavior? To do this, we need to dig deeper into the data, and we can apply the same integrated volumetric approach that I just showed on the previous slide, but now apply that to the stability and repeatability data that we've collected, where we do see these very significant differences. Again, this is a very abstract and unusual measurement. So the vertical axis on these graphs, really the numbers are not important, but again, what matters is how we see variations. When we're looking for equivalence between different products, we expect that these measurements ought to follow the same trend and follow the same values, because we've normalized them by the performance of each particular device. So when we're looking for equivalence, we expect the lines to be the same. It gives us, in a way, a score for plume repeatability and equivalence. And what we can see is when we analyze the temporal stability, how much we get variation over time in the plume, we can see that the red and the blue lines for our new low GWP propellants both lie below the black HFA 134A. And that tells us that both of these are showing reduced stability overall, very close to the orifice. And we see that these profiles all tend to diverge away from the orifice. On the right-hand side, we're looking at the shot-to-shot repeatability. And we see a very different story here. Now, when we look at the shot-to-shot variations, and we see that here, the variations really occur further downstream in the mouthpiece. All the lines move together until the point at which the spray plume becomes wide enough to interact with the inside of the mouthpiece wall. And at that point, they then diverge. And this gives us a clue. This tells us that if we want to try and tune the stability of the spray, we need to go right back to the orifice, because that's the point at which these start to diverge. But if we want to have some control over shot-to-shot repeatability, and perhaps looking at mouthpiece actuated deposition and trying to manage that, adjustment of the mouthpiece is where we really want to be focusing our efforts in those regards. And so this is just one example of how this big data approach to analyzing very large numbers of spray events with some more complex statistics can start to give us clues as to how we can improve product performance. All the results I've shown you in the last few slides have been for solution formulations, and we might then ask the question, well, do suspension formulations behave differently, or will the same things hold true? And so far in the data analysis that we've done, we see exactly the same trends for suspension formulations as we do for solution formulations. Again, here the orange lines and the blue lines for our 152A formulations and our 134A formulations lie on top, above the HFO 1234 formulations, which gives us lower scores. And this tells us again that the trends are very much the same for suspensions, even though the values and the actual average structure of the plume may change. So then this brings us to a question of whether we can then tune spray performance based on the knowledge that we gain from these measurements that we've done. So our current work is focused on tuning performance by focusing on, for example, varying the orifice. Now, the orifice diameter is something that we generally don't want to modify too much, because it's connected to the metering valve volume, formulation properties, discharge rates. And so if we want to keep the orifice diameter at its appropriate design size, the other variable that we can do is we could control orifice length. And so we've been undertaking a computer simulation-based study where we can run very large numbers of simulations repeatedly of inhaler sprays and look at what happens if we tune and modify the orifice length. What this does is, by increasing the orifice length, it increases the amount of time that the formulation spends in contact with the orifice wall. This increases residence time for phase change and heat transfer, and it alters the condition of the fluid as it exits the orifice, and it's going to change that chaotic atomization and spray breakup and flashing process. And on the right there, you can see some early results from our computer simulations, which suggest that as our data analysis of the sprays from the in vitro studies predicts, by increasing the nozzle length, we can tune the spray width, we can bring it down. And so this gives us another dial that we can turn in the design process by making very small adjustments in orifice length. We can make small adjustments in the spray width and tune the width profile of the spray to bring it into equivalence with another product, for example. This is not the only way in which we can adjust spray performance. So I'll hand back to Ben now to talk about some other routes that we can take.
Thanks, Sam. We've seen from previous literature that the vapor pressure of binary mixtures of propellant and ethanol behave non-ideally. But we didn't know at the start of this work what the behavior of the new propellants, the low GWP propellants in binary mixtures with ethanol, we don't know what the behavior would have been. So we've undertaken a campaign of measurements to characterize varying ethanol fraction across a range of different temperatures, what the vapor pressure of the formulation is. And we can see that the vapor pressure does indeed reduce, as we would have predicted, with the addition of ethanol in the formulation based on our previous knowledge. But what was interesting to find was that the reduction of vapor pressure was less severe than for ethanol 134A mixtures. And it does invite the possibility that within the bounds of formulation and solubility constraints that we're working, we may be able to adjust the level of ethanol in a solution-based product, tuning that concentration to aid the performance of our products also. So, summarising and concluding the work that we've presented today, we know that there are property differences of the propellants, and we've shown that these property differences lead to pharmaceutical performance differences between products when we just do a straight switch of the propellant in that formulation recipe. But the differences are subtle, and we can explain some of the performance differences with the propellant property differences, the thermodynamic and the physicochemical properties. We've used a range of new and novel techniques that have unlocked new understanding and gained further knowledge to enable us to mitigate these performance changes. And really, the performance changes are small enough that we can tune the actuator, the hardware, and maybe even the formulation to gain an equivalence of performance between our current propellant formulations and a pMDI format and the new low GWP propellant products. We may choose to do that by tuning the orifice length. Lengthening the orifice, we've shown, narrows the plume width. And this may be one way in which we can either change the plume width or the mixing of the propellant formulations when they're atomized. We may also be able to change the ethanol co-solvent. We've seen that the vapor pressure of these propellant formulations with low GWP propellants are less sensitive to addition of ethanol than the 134A binary mixtures were in the past. Our ongoing work is exploring how hardware optimization can be used to mitigate the performance differences. And so we're undertaking a series of experiments currently, changing the actuator geometry and mapping the effect of orifice size and length changes. And that will be coming out in the literature in the coming months. And so with that, I'd like to thank everybody for joining our webinar today, for your interest, and we turn it over to some questions now.
Well, thank you both very much for that insightful presentation. I'd like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, I've already received some questions for you both, so we'll get ourselves started with those. The very first question here would like to know what would be the potential impact on patients with a shift to green propellants?
Thanks, Ryan. I think I'll take that question to start off with. I think our ultimate aim would be, and with the work that we've undertaken here, and we presented some of this today, is that the patient really sees no difference. When we change the propellant gas in the pMDI product, we want the patient to see no difference in the product performance to the current propellant products that they have today. So really, they would see no difference. If we don't make the switch to the green propellants, then ultimately the propellant pMDIs may diminish on the market and will become non-viable. And that would mean a patient would have to switch to an alternative device platform. That could have many different impacts on their compliance, on their clinical outcomes, and of course we want to avoid that. So to us, it's really we don't want a patient to even realise that the product has changed, and I think we're on a route to be able to achieve that.
All right, very good. Thank you so much for that. The next question is curious, what is the difference between the extinction and the standard deviation profiles?
I'll be happy to take that one, Ryan. Thank you. So in our analysis, where we talk about the light extinction, we're talking about an average. So when we collect hundreds of spray events over a very large number of points in time, the extinction profiles that we've shown in this presentation are the average. That's the expected value. If we randomly sampled a point, what would be the most likely value that we would see in terms of the amount of light scattered by the droplets and particles. This tells us the average structure. The standard deviation tells us by how much the amount of light scattering tends to vary around that mean value. And what we're hoping to see is that standard deviation is neither too large or too small. Even if the mean profile, that is the extinction profile, is symmetric and it looks very good, if the standard deviation is too high or too low, that tells us that there is simply too much variation over time or between actuations around that mean value. And so it's actually that standard deviation value that is almost more important to us in our analysis and gives us more information.
All right, very good, very insightful. Thank you for that. The next question here is curious about how does an increased interest in green propellants relate to other trends in inhalation delivery?
I'll take that question again. Thank you, Ryan. I think ultimately socioeconomic drivers are raising the awareness to everybody of carbon footprint or their emissions and their use of resources. So really, this is driving the patients to want to have more environmentally friendly products, and that's what we're working towards. We're trying to achieve that. There are alternatives out on the market, but they may have other issues in terms of fossil resources, marine eutrophication, and other impacts on the environment.
All right, very good, thank you. Next, someone's wondering, what is the effect of nozzle length on APSD using an ACI or NGI?
So I think at this stage, we have some knowledge on this area. We know that changing the length by small amounts allows us to relatively speaking, maintain an APSD profile. We need to explore how much further we can push the changes on orifice length to understand how much that's going to impact on APSD. But we're in the early stages of characterising that at the moment. We're seeing some promising results so far.
All right, thank you. Next question here asks, what are the biggest gaps in the current understanding of low GWP propellants, and how can the industry address those gaps?
Again, I'll jump in here at this point. There are obviously a range of technical considerations that we've discussed today. We are in the infancy of the low GWP propellant switch. We are seeking to understand and characterise the performance of these pMDI products as we've discussed today. One of the big areas we will have to understand over the longer term is the physical and chemical stability of products as they approach a 2-year shelf life. And the more time that passes, the more we will understand that. Of course, there are other considerations. There are the regulatory considerations. What requirements exactly are going to be on new products? clinical implications, we don't fully understand yet, and they're not fully mature in the industry and in regulation what clinical requirements are required. So I think that there are quite a lot of sourcing would be another potential problem. We know that we do not fully understand the tox profiles, but those are coming very quickly. Cora particularly have done a lot of work on 152A, Honeywell working on 1234ZE. So we're really optimistic on the challenges that we face. There, we're making good strides forwards to enable low GWP propellant products.
All right, thank you very much. Now, Ben, I'm wondering if you could maybe just speak a little generally about the tools used for modeling and simulation.
Well, we have a wide range of tools available to us at Kindever and via the collaboration we have with Daniel at Monash University and the Woolcock Institute. We have a range of kind of novel characteristic and capabilities that are unique to Kindever based on the work that we've put in over the past few years into developing these new capabilities alongside our understanding of low GWP propellant sprays. So yeah, we have computer modeling capabilities to be able to predict particle size distributions, spray plume properties, both internally and Dan has them himself as well. We have novel measurement capabilities for plume temperatures, temperatures inside the device, the actuator, pressures inside the actuator. And all of these experimental techniques have enabled us to feed the knowledge that we've gained in the lab back into our computer simulations, much like the way that Dan's experimental X-ray measurements over in the US have contributed to the modelling work that he's undertaken. So yeah, we really like to employ that novel capability and novel technique in tandem with our pharmacopeal measurements, so that we can really get a richness of data to understand, the impacts that we see, or the effects that we see in our pharmacopeal tests. So we can delve deeper into the physics and our fundamental understanding to enable, accelerated pMDI product development and better future outcomes, either that be optimizing for really efficient inhalers for new products, new entities, or for essentially generic type products where we kind of match the performance of current products that are on the market.
Wonderful. Okay, thank you so much for that answer. The next question here is wondering, so based on all the data collection, what is the benchmark for equivalence as the methods developed are more detailed than Proveris or Oxford laser?
Dan, do you want to answer that, or shall I have a go?
Oh, sure. I'm happy to speak to that one. It's important to point out that some of the optical techniques, such as those shown here that we're developing, are really very different to the conventional spray pattern and plume geometry type measurement technique. So as we move closer and closer to the orifice, things get smaller and smaller and move faster and faster. And so our technique is optimized for very in-close measurements. And so therefore the way in which we're going to be, for example, developing tools to measure equivalence of the spray plume in this region are going to look very different to what we might have, for example, from a spray pattern and plume geometry measurement much further downstream. So at this point, what we're looking at really is a very measurement capabilities that define equivalence in our experiments based on a very fundamental relationship with the way light scatters from particles. So if we compare that, for example, to a conventional spray pattern plume geometry measurement, which might be more suited for the quality assurance type purposes, that you're going to be getting, it's really apples and oranges. So it's a completely different technique that perhaps is better suited to the design and exploration phase of the development.
Very good. Thank you very much for that insight shared for that question. Next up, someone's wondering which propellant is preferred as to providing a match in pharma performance of albuterol sulfate suspension product.
Thanks, Ryan. I'll take that question. I think from the work that we've undertaken so far, I don't think there is a preferred propellant from a technical perspective. I think we could potentially use either. we've shown that the differences that we see when we purely switch the propellant in the recipe are really, we can mitigate those differences by tuning of the actuator. So I think that we can overcome the differences that have been seen when we compare to a 134A current product. I think the big difference is we have to ensure that we handle the product in an appropriate manner. And yeah, I think we could use either.
Wonderful, thank you. Now, I wonder if a company wants to reformulate their inhalation products to improve sustainability, where should they start? What does this process look like?
So I think first off, the company or the person would want to understand what their product performance profile that they desire would be. There is a range of different propellants, as we've seen today. There will be a range of different hardware and componentry that the product could make use of. So we really need to obviously understand the space that we're operating within, and that will be a compatibility study after an initial viability study. And we really go through the kind of normal formulation development routes that we've employed over many years at Kindeva. We have a lot of experience in developing the pMDI products that you're talking about. So it really is a kind of a standard approach to the product development, really. We're not overcoming problems or challenges that we're not used to or not familiar with, even when we switch to low GWP propellants.
Wonderful. Thank you very much for that answer and for all of the answers today. However, we have reached the end of our Q&A portion for the webinar. Now, if we couldn't attend to your questions, know that the team at Kindeva will follow up with you, or if you have further questions, you can scan the QR code on the screen to continue the conversation with our speakers. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, there's a link to view the recording of this event in the chat box, which you can share with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks, Ryan. Thanks, folks.